our YouTube channel, Mixed Clinical and Research Laboratories. Today, I will share brief introduction about PCR, which is also known as polymerase chain reaction. So without wasting time, let's get started.
What is PCR? Polymerase chain reaction. Basically, it's an in vitro technique that takes specific sequence of DNA of small amount and amplifies it to be used for further testing. Chemical components of PCR technique are magnesium chloride, buffer, deoxynucleotide triphosphate, primers, and enzyme DNA polymerase. Now I would like to share basic requirements for PCR reaction. DNA sequence of target region must be known, and the primer should be typically 20 to 30 bases in size. These can be readily produced by commercial companies, can also be prepared by using a DNA synthesizer. Further on, we need thermostable DNA polymerase. For example, Taq polymerase, which is not inactivated by heating to 95 degrees Celsius. We need a DNA thermal cycler, a machine which can be programmed to carry out heating and cooling of samples over a number of cycles. Now I would like to share principle of PCR, which includes its purpose, conditions, and components. What is the purpose of PCR technique? PCR is used to amplify a lot of double-stranded DNA molecules with same size and sequence by enzymatic method and cyclic condition. Now further on, moving towards conditions of PCR technique, which includes denaturation of double-stranded DNA template, annealing of primers, and extension of double-stranded molecules. First step of PCR technique is denaturation. This is carried out at temperature 92 to 94 degrees Celsius. In this step, double-stranded DNA melts into single-stranded DNA. Second step is annealing, which is carried out at temperature 50 to 70 degrees Celsius. In this step, primers bind to their complementary sequences. Next step is extension. This step is carried out at 72 degrees Celsius temperature. In this step, DNA polymerase enzyme binds to the end annealed primer and extends DNA at the 3' end of the chain. In last step of PCR technique, all the steps of PCR like denaturation, annealing, and extension are repeated or cycled many times to amplify the target DNA. The number of cycles is normally carried out 25 to 35 times but may vary upon the amount of DNA input and the desired yield of PCR product.
I hope that this video will be informative and beneficial for you. So don't forget to like this video, subscribe our channel Mixed Clinical and Research Laboratories, and don't forget to press the bell icon so that you can receive the notification of our latest videos. Thank you so much.